Guys, in this video, we're going to start using those terms we learned in our first video and how to apply those. And the title of this video is Monohybrid Cross. Well, let's talk about what that means first of all. By the way, I also jotted down a couple of definitions that we went over in the last chapter. So if you want to pause the video and copy these down, we're going to talk about these again. Okay, are you all caught up? So monohybrid cross, you guys have learned that mono over the course of the year means one. And hybrid, we're talking about traits. So when we do a monohybrid cross, we're looking at the inheritance of one trait. So we have the inheritance of one trait. And in this one, we're going to look at eye color in humans. We're going to look at brown eyes versus blue eyes. Well, let's say dad has brown eyes and mom has blue eyes. Now, one of the first things you're going to see is that we use letters to represent the alleles. So our alleles are represented by letters so that we can kind of have something to hang on to when we are looking at this inheritance. So we always then ascribe a trait to a letter, and we usually use a capital and a lowercase. The capital is dominant and the lowercase is recessive. So in this case, we're going to say that capital B is a brown eye allele and lowercase b is a blue eye allele. Brown eyes are dominant to blue eyes. So we have dominant, we have recessive. And remember, when we say allele, we're talking about a gene. We're talking about a section of a chromosome that has the code to build a protein that's going to make eyes either brown or blue. So that was a whole lot of terminology there, but that's review from the last video. Again, a gene is a section of a chromosome that codes for a protein. And dad has a one allele is for brown eyes, and we're going to say he is homozygous, or I'm sorry, he's heterozygous. We're going to say he's BB. So father is heterozygous. And if mom has blue eyes, what does her genotype have to be? Two lowercase b. So that's the only way you can have blue eyes. So back when we did meiosis, we saw that when gametes form, so if this is dad's genotype and this is mom's genotype, they each have, this is on a pair of homologous chromosomes, and this is on a pair of, they would be on the same one, like maybe it's number 15. I don't know the number. But one of the homologous pairs would have a allele, capital B, and the other would have a lowercase b. So think about it this way. If this was dad, this is dad's chromosomes and these were mom's chromosomes. At that same spot, they would have their alleles. This should be longer because these should be identical. Sorry. And dad's would be a capital B, lowercase b. Lowercase b, lowercase b. Again, these are the genes. These are sections of the chromosome. Well, when we go to form gametes during meiosis, so remember we form gametes during meiosis. The law of segregation says that these, the gamete, you're going to get one from mom and one from dad. And it's going to be completely random whether you get this one or this one, and whether you get this one or this one. So the law of segregation tells us that the inherited gamete, the inherited gene from the gamete is it's completely random. So it is a random. The law of independent assortment tells us that this allele isn't going to affect this allele, and this allele isn't going to affect this allele, that there's no connection between the alleles. There is no connection between the alleles. So to sum it up, what these two laws are telling us is that the inheritance of a trait, and this is the ideal. Now we're going to find out that the law of independent assortment and segregation doesn't always hold true. But What this means is that there should be a complete random inheritance. Or probability. So I just introduced the P word. At its essence, genetics is a study on probability. And if you take a statistics course, they'll talk about a lot of different ways to look at probability. Well, one of the ways that we could analyze probability, so one of our probability predictors is something called a Punnett square. I think it has two T's, I'm not sure. Now, there's other ways you could do probability. There's mathematical calculations, but in biology, we usually use Punnett squares. So what we will do is we will line up the alleles for one parent on, we'll make this box. And for a monohybrid cross, we need a 2x2. So I'm going to create a box here. I'm going to make it 2x2. So we're going to put, I usually put the female alleles at the top and the male alleles here. It doesn't have to be, but it could be. Again, that's just kind of my tendency. And then each of these sections, this section, this section, this section, and this section, the labels are the alleles. So, whoops, yep. Yeah, so we have the female is BB and the male is heterozygous. What we do then is we do, if you've ever played Battleship and you talk about like A5 and you go over A and you come down 5, we're going to pull, I'm going to color code these. So I'm going to make Dad's purple. We'll write Dad's alleles. So we'll bring those across this way and we'll bring Dad's here. And then we'll draw Mom's down. Let's do her in pink because those are pink. So we'll draw hers down. So she'll be B. So here's what this means. If I have 4 offspring, the way that I'm going to read this Punnett square is if I have 4 offspring. And by the way, we represent the breeding pair or the parental pair. So if we're talking about flies, you don't really have parents, as the P1 generation. The offspring, which in Latin is filial, is going to be the F1 generation. So if I have 4 offspring, 2 of the 4 will be heterozygous. Y-G-O-U-S. BB, which gives you brown eyes. 2 of the 4 will be homozygous recessive. Blue eyes. So if you guys know your math, you know that if you take 2 divided by 4, that's going to give you 50%. So 2 divided by 4 is 50%. So if you cross a heterozygous brown-eyed father with a homozygous recessive blue-eyed mother, you would expect half of their kids to have brown eyes and half of their kids to be blue eyes. Now, if I flip a coin, I have a 50-50 chance of having heads or tails. So does that mean if I flip it twice, I'm always going to have 2 heads or a head and a tail? No, sometimes I get 2 heads, sometimes I get 2 tails. So what you have to remember in this is this key word, probability. This is probability. Just because I predict that this is probably what's going to happen doesn't mean that is what's going to happen. This is a probability. So what I want to do in the part 2, this is part 1, I want to go over some practice problems. So let's do a quick recap. In meiosis, we've talked about in meiosis that the chromosomes separate independently. You have as much of a chance of getting this from dad as you do from getting that from dad, and that these two chromosomes together won't affect the allele here and the allele here. They're not connected in any way. There's no greater chance of getting this or this. This gives us a complete random probability. So because of that, we can use this tool called a Punnett square. And again, we put mom's alleles on one side and dad's on the other, and we can find this probability. So, and again, keep in mind, it is a probability. So let's look at some sample problems in part 2. So I'm just going to write a part 1 up here. Part 1. Okay, on part 2.